It's my pleasure to introduce Dr. Gina Peloso, a friend and colleague for a long time. Gina is a lifer essentially at BU. She did her undergraduate there in biology and then her PhD in biostatistics. Did leave BU a little bit to go over to the Broad Institute and Harvard Medical School for her postdoctoral training, which is where we met when I was a postdoc myself, and is currently an associate professor in the Department of Biostatistic at BU at the School of Public Health. Gina's work focuses on uh, statistical genetics and genetic variation of blood lipid levels. She's a leader in many consortia for the genetics of blood lipids, and I think I imagine she'll probably talk about that a little bit today. And on a personal note, Gina and I have collaborated since we were postdocs, and she's been helping me when I started my lab at UCSD. So, Gina, take it away.
Well, thank you so much for that warm introduction, and thank you so much for the opportunity to share some of my research here with you today. So my overall research objective is to leverage large-scale human genetic data across the allele frequency spectrum to try and make insights into complex traits, particularly around blood lipid levels in the population. So in this presentation, I'm going to review what we know about the genetics of blood lipids in the population using genome-wide association studies. And then I'll spend the majority of the time talking about what we're doing to determine the contribution of rare non-coding variation to blood lipid levels using whole genome sequencing data. So we're on the same page. Conventional blood lipid levels include LDL cholesterol, or your bad cholesterol, HDL cholesterol, which is considered your good cholesterol, triglycerides, as well as total cholesterol, which is a sum of your other cholesterol measurements. And we call these good and bad cholesterol because of their epidemiological associations with coronary heart disease. So in epidemiological studies, as your non-HDL cholesterol, which is a function of your total cholesterol minus your HDL cholesterol, and it's highly correlated to your LDL cholesterol. As you increase those measurements, your risk for coronary heart disease increases. And likewise, for HDL cholesterol, as you increase your HDL cholesterol, your risk for coronary heart disease decreases. So there are a couple of characteristics of blood lipid levels that I think make them exemplary traits for genetic analyses. So all these lipid measures are heritable risk factors. They have approximately about 50% heritability estimates. They're continuous. They're biomarkers that are routinely measured in the clinic to predict risk for heart disease, and they're targets for therapeutic interventions. Now, genome-wide association studies are typically used to try and identify common single nucleotide polymorphisms that influence a trait. They're particularly useful with complex traits, which are traits that have both a genetic and environmental components. And when there's not necessarily a single gene or a set of genes that are responsible for the trait, and the genetics for underlying the traits may be very subtle, so small effect sizes. And so when we do a GWAS, what we're getting out of that is millions of association results, right? So we're getting each genetic variant and whether it's associated with our complex trait of interest, like LDL cholesterol. So we're getting an effect size for that and a p-value for each of the genetic variants we're studying. and we're getting millions of these results. The latest Global Lipids Genetic Consortium, GWAS, looked at 5 blood lipid traits and tested about 52 million variants with minor allele counts greater than 30. This included 201 cohorts across a diverse set of populations that implemented this standardized analysis plan to perform linear mixed effects models. And this led to a meta-analysis of up to 1.65 million individuals. And while most of the individuals are of European ancestry, with this effort, we substantially increased the sample sizes of the non-European studies contributing to our GWAS for lipid levels. And from this meta-analysis, we identified 2,286 index variants that were associated with at least one of the 5 lipid traits that we studied. And what I think doing these large-scale GWAS worth doing is not the identification of loci associated with a complex trait, but our ability to do secondary analyses that perform better when we have robust GWAS summary statistics that are based on larger sample sizes, more diverse samples that are contributing to the GWAS. And to make that point a little further, I'm going to point out some of our main findings from our GWAS of lipid levels. So first, we used our GWAS summary statistics to develop polygenic risk scores, right? And a polygenic risk score is just a weighted sum of genotypes, and we're weighting by the effect sizes from our GWAS. And so here we created polygenic risk scores for using all individuals in our meta-analysis, which is these dark blue plot points. And then we created polygenic risk scores using the weights from population-specific meta-analyses. And then we tested those scores in individuals from various studies around the world that did not contribute to the meta-analysis. And what we saw was that our polygenic risk score, right, for LDL, it was most informative when it used that multi-ancestry GWAS, so using all individuals. And that multi-ancestry polygenic risk score, it performs equally as well in the European ancestry Americans, your African ancestry Americans, as well as these continental African individuals compared to ancestry matching, right? The polygenic risk score to that particular population. And so what this means is that you do not need to ancestry match your polygenic risk score to your sample. You can use this multi-ancestry GWAS for your weights for your polygenic risk score. And so it's not clear, this works really well for lipid levels. It's not clear whether this result extends to other complex traits that might have different genetic architectures across populations. However, for lipid levels, right, not having to ancestry match your GWAS weights for your polygenic risk score to the sample is beneficial because humans don't typically fall into kind of discrete categories. We next use our polygenic scores for lipid levels to identify relationships of genetically predicted lipid levels with other diseases and conditions using a PheWAS approach, a phenome-wide association study, and we leverage the UK Biobank and MVP here. So along the x-axis here, I have disease classes. And each point in this figure is the association between our LDL polygenic score and each of the disease phi codes. And the y-axis is the minus log 10 of the p-value, so giving strength of association. And as expected, we see that genetically predicted LDL is associated with mixed hyperlipidemia and coronary atherosclerosis. However, we also see some interesting associations between here bilirubin and cholelithiasis, or gallstones. And there's a contradictory epidemiological data on the association between lipid levels and risk for gallstones. However, we found that extreme lowering of LDL cholesterol may impact rates of gallstones. We also looked at sex-biased lipid loci. That is genetic variants with different effects in males and females. And we showed that 3 to 5% of the autosomal lipid-associated loci demonstrate these sex-biased effects. And this gene UGT2B17 is one of our 10 sex-biased loci that also has a sex-biased EQTL effect. This gene is implicated in androgen and drug metabolism. And the lead variant in this gene shows a significantly stronger effect on LDL cholesterol in males, right? It co-localizes with a male-specific liver EQTL associated with increased expression of the gene. And we saw that it's associated with testosterone levels in males in a sex-specific PWAS in UK Biobank. Using our GWAS results, we also tried to determine regulatory mechanisms underlying our lipid associations by trying to integrate multiple layers of functional data, including Capture-C data to identify functional genes. So I have this LDL GWAS loci here, and this loci has an EQTL for the gene RRBP1 in the liver, which is the relevant tissue type. And variants in the credible set for this loci interacts with the gene promoter in both adipose and liver tissues in high C data. And this interacting variant is also in an open chromatin peak in 3 liver-related cell types. So we're trying to kind of dig into our GWAS results by trying to learn the regulatory mechanisms of our associations. The Global Lipid Genetics Consortium, we release all of our summary statistics for others to use, hopefully widening the impact of doing a GWAS. And these results that were published a couple years ago have been used in multiple studies, including for Mendelian randomization, kind of overlap with other traits, creating polygenic risk scores. We also created a browser displaying our gene prioritization and PWAS results. And lastly, I just want to share our general Global Lipid Genetic Consortium website. Now, GWAS focuses on single nucleotide variants, where we're looking at each variant individually for association with some outcome. And we typically can do GWAS with what we say are common genetic variants, and how we define what common is dependent on the sample size, right? So as sample sizes have increased, we can go further and further down the frequency spectrum and look at rarer variants. And what I tried to argue over the last several slides is that what I think is most useful about doing GWAS is the ability to do fruitful secondary analyses using computational approaches. I think one of the limitations still is being able to connect genetic variants with functionally and relevant genes and mechanisms. And while we are trying to make inroads on that, it still requires looking deep within each of the loci to be able to fully understand the mechanism behind the associated variants from a computational point of view. So we also now look at exome sequencing. Exome sequencing focuses on the coding regions of the genome. And I think one of the major advantages of exome sequencing is interpretability. So because coding variation leads to changes in the protein structure. However, you're only looking at less than 2% of the genome. So there's a limited portion of the genome that you're querying when you're doing exome sequencing. Though identifying rare damaging coding variation has helped to elucidate biological pathways underlying lipid levels and atherosclerotic diseases such as coronary artery disease, and there are several successes in identifying rare damaging coding variants in these genes here that have led to novel therapeutics to modify blood lipid levels and lower risk for atherosclerotic diseases. A couple years ago, we performed a gene-based association test by aggregating rare variants in genes from over 170,000 diverse individuals from multiple studies at that time. And we found 35 genes that met our exome-wide significance level. And so along the x-axis here is our 35 genes. And along the y-axis is the traits we tested here. And the genes that were associated with each lipid trait are identified through these dots. And as expected, given the correlation between the lipid traits, we see oftentimes more than one lipid phenotype is associated with each of these genes. And most of the associations that we found were actually with known lipid genes indicated by the yellow highlighting, as well as those 3 genes that I mentioned at the onset, which is ApoC3, NPC1L1, and PCSK9. Using that results, we wanted to next test the utility of sets of genes that showed some evidence for association, but did not reach a stringent significance threshold. So here we looked at Mendelian genes, right? Genes identified as drug target genes, as well as GWAS loci, categorized on their distance from the index SNP and whether that index SNP was a coding SNP or a non-coding SNP. And as expected, we found that Mendelian genes and LDL drug target genes, right, showed an enrichment in association compared to a matched set of control genes. But what was interesting is we also observed that the closest gene, whether that closest gene was a GWAS coding variant or a non-coding variant, right, that also showed an enrichment of association compared to a matched set of control genes. So the main takeaways from our exome sequencing study was that we were able to discover genes associated with blood lipid levels, right? We were able to replicate well-known lipid-associated genes, as well as have some new gene-based associations for novel genes with lipid levels, which can indicate new genes that can be discovered through exome sequencing, particularly with these large sample sizes. We also determined whether we had rare variant coding associations within genes, and can we prioritize those genes? And so what we found is with, in addition to Mendelian lipid genes and LDL drug target genes, genes closest to the GWAS index variants are enriched for gene-based associations. And so this provides evidence that the closest gene to the GWAS index variant is a functional gene in the region. So the next piece we started looking at is whole genome sequencing data, right? With whole genome sequencing data, this allows us to look at rare and ultra rare variation that might be unique to a population, unique to an individual or family. And when we're looking at rare genetic variants, we can't test them individually for association with our outcome. So we have to use aggregate-based association testing, as we did with the exome sequencing study. However, what is unique to being able to look at whole genome sequencing data is our ability to look at rare non-coding variation, which is not accurately captured with GWAS or exome sequencing. So there's an increasing number of studies that have generated whole genome sequencing data for complex traits. We started looking at the whole genome sequencing data from the NHLBI Transomics for Precision Medicine program. This program, TopMed, integrates whole genome sequencing and other omics data with molecular, behavioral, imaging, environmental, and clinical data. TopMed now has over 200,000 participants from over 85 studies. However, this was opportunistic sequencing. So the contributing studies have various phenotypic focuses, and not all individuals that have been sequenced have collected blood lipid levels. However, what I think is great about TopMed is they made a concerted effort to sequence individuals from diverse ancestries. So over 60% of the data is of non-European ancestry. So our first analysis with the whole genome sequencing data was using 16,000 individuals that were available at the time with both blood lipid levels and whole genome sequencing. And with this data, we looked at common variation individually for association with lipid levels, rare coding variants, non-coding rare variants, as well as the genetic influences on hypercholesterolemia. So we were able to study the association of aggregates of rare non-coding variants for the first time using this data. And what we did at this point is we employed 4 grouping schemes for our rare non-coding variants. First thing we tried was a sliding window approach that tiles across the genome at fixed widths and only includes variants that are non-coding annotations that are consistent with promoters, enhancers, and DHS sites. We also tried 3 approaches where we tried to map non-coding variation to discrete genes as the functional unit. And so we mapped variation in promoter and enhancers and DHS sites near a gene's transcription start sites. We looked at promoter enhancers and the concurrence of enhancers and DHS sites with HepG2 expression. And then we looked at histone marks within high-seq data contact regions mapped to genes. And using these approaches, we did not find any rare coding associations detectable in this study due to the low sample size. Now, based on genetics, there's 2 routes to get to a high LDL cholesterol and increase one's risk for coronary heart disease. So you can have a single variant, right, that drives a person to a high LDL. And these variants are typically very rare in the population and are protein coding, but they have a large effect size. And we have several genes for lipids that we know are monogenic for lipid levels. On the other hand, right, you could have many genetic variants that incrementally increase your LDL just a little bit, but in aggregate lead to a high LDL cholesterol. And this is your polygenic risk, and it can be assessed through a polygenic risk score. And whole genome sequencing data allows us to actually simultaneously assess both these rare coding variation in these monogenic genes, as well as the polygenic component to high LDL cholesterol. So we assess these 2 components in our data. And first, you can see we saw a very small number of individuals that had a monogenic variant leading to a high LDL. And if you had one of these monogenic variants, right, you had a substantial increase in your LDL cholesterol, substantial odds for developing high LDL. However, like we have a larger proportion of individuals that have a high polygenic risk score that also leads to a higher odds of having high LDL cholesterol. And this indicates we could identify more individuals with a high LDL cholesterol using a high polygenic risk score compared to just looking at these monogenic variants. All right, so as more data was sequenced, we were able to extend our analysis to the next phase, and we were able to look at 66,000 subjects with whole genome sequencing and blood lipid levels within TopMed. And so here we were able to perform an analysis of 428 million variants, of which 400 million of them were not priorly assessed for association with lipid genetics. And as we learned with our previous TopMed project, for rare non-coding variants, it's really not clear what the appropriate annotations that we should be using in these aggregate-based set tests. So here we leveraged the STAR framework that was developed within the TopMed Lipids Working Group. This is a scalable and a powerful variant sets and tests that uses an ominous multi-dimensional scaling weighting scheme to incorporate both qualitative functional categories as well as multiple in silico variant annotation scores for genetic variants to get a p-value for an aggregate set of variants. So instead of using just one set of annotations, right, we are able to incorporate these 12 annotations into our tests. And using this STAR approach, we observed several genes associated with blood lipid levels, both coding aggregates as well as using non-coding aggregates, and non-coding aggregates linked to genes. Importantly, we identified rare non-coding variants in proximity to Mendelian genes, specifically LDLR and PCSK9. And so those also contribute to the genetic basis of hypercholesterolemia. So in addition to monogenic coding variants contributing to hypercholesterolemia, we have polygenic, right, common variants that contribute to hypercholesterolemia. And now we're seeing that we have these rare non-coding variants in LDLR and PCSK9, right, contributing to hypercholesterolemia. And this speaks to the influence of the spectrum of variation in these loci associated with high cholesterol levels. This matters because only a small fraction of individuals with severe high cholesterol have what's called a hypercholesterolemia, familial hypercholesterolemia variant in one of these three genes, LDLR, ApoB, or PCSK9. And the presence of one of these variants in one of those genes is considered independently prognostic for coronary artery disease beyond one's lipid levels and merits the consideration of more costly lipid-lowering drugs. So we observed that there's these rare variants in the introns of LDLR that lead to these large increases in LDL cholesterol. And these effects are approximately the same effect as is observed with clinical FH variants in LDLR. So we argue that a whole genome sequencing approach to lipids disorders will markedly improve the diagnostic field beyond just the existing limited approaches here. Next, we investigated a particular class of understudied variation called long non-coding RNAs. These are transcripts greater than 200 nucleotides in length that biochemically resemble messenger RNAs, but do not code for proteins. They don't bind to common protein complexes, making them complex to determine their function. The function of only a few of them are understood. However, it's thought that they're potentially involved in lipid metabolism in humans or mice. So what we wanted to do was leverage the whole genome sequencing data and rare variants to determine whether long non-coding RNAs contribute to variation in blood lipid levels. So what we did was we integrated 4 different sources of long non-coding RNAs, and we leveraged the TopMed data and did analyses for these sets of long non-coding RNAs within TopMed against blood lipid levels. And so we did this association using the STAR framework. And then we wanted to determine whether our associations we observed were distinct from common variants identified from GWAS, as well as coding variants. And we also performed replication using whole genome sequencing. And we tried to connect the long non-coding RNA signals that we observed from whole genome sequencing with the long non-coding RNA expression levels. And so at a corrected significance level, we identified 83 long non-coding RNAs associated with blood lipid levels through using whole genome sequencing. However, all of our associations were in known lipid loci. So to determine whether our long non-coding RNA associations were distinct from our GWAS associations, we performed conditional analysis. So just to orient you here to these plots, along the x-axis is the p-value for the association between the long non-coding RNA and a lipid. And then here along the y-axis is the minus log 10 of the p-value after adjusting for here in the first plot GWAS variants, non-synonymous variants, synonymous variants, and loss of function variants. And so what we saw was that some of our long non-coding RNA associations dropped off in significance after adjusting for GWAS variants, as well as adjusting for rare coding variants. However, several of the associations seemed to be distinct. They weren't changed after doing the adjustment. Overall, this led to most of the long non-coding RNA associations being distinct from the GWAS variants and the coding variants. We then tried to replicate our associations in the UK Biobank using approximately 139,000 individuals with whole genome sequencing and blood lipids that were available at the time. And we were able to replicate a little over half of our associations. There's a couple of reasons why we may not be able to replicate our rare variant associations. For one, you have to observe the variants in both the discovery and the replication studies. And by definition, when we're looking at rare variants, they're not seen very often. And secondly, TopMed, our discovery cohort, is more diverse of a study than UK Biobank. So we may not be seeing the variants we see in TopMed in UK Biobank due to differences in the ancestry composition. So actually being able to replicate over half of our associations, I think doing a rare variant test is promising. We next tried to look at the expression of long non-coding RNAs. So we leveraged about 2,000 individuals in the Framingham Heart Study that had RNA-seq data, and we overlapped our significant lipid-associated long non-coding RNA genes from the whole genome sequencing analysis with the Framingham RNA-seq data. We limited to the gen code link RNAs that were available. and we observed 4 gene expressions associated, where 3 of those were for just 1 link RNA against 3 lipid traits, and that is an antisense of Nectin-2. And so what we see here is we saw that rare variance in this Nectin-2 antisense, right, was associated with lipid levels. We also saw that the expression of the Nectin-2 antisense was associated with lipid levels. However, we weren't able to assess whether rare variants in the long non-coding RNA variants, genetic variants, were associated with expression levels due to the low number of individuals with the expression data and having to observe the genetic variants in those individuals. So you need large number of individuals to do rare variant testing, and we have a limited sample size with the expression data. So there's many limitations of looking at long non-coding RNAs. I think some of the limitations include most are considered transcriptional noise. We tried to use, what we're trying to use is like association analysis to see which ones might be relevant for lipid biology. There's a lack of annotations for long non-coding RNAs. However, our STAR framework that leverages multiple in silico functional annotations helps get around this. There's a lack of transferability of information across link RNAs. So if you know what one does, it doesn't necessarily translate to the function of another one. And they're lowly expressed. So there's not good expression profiles for long non-coding RNAs. However, some long non-coding RNAs will have functional consequences. And our research suggests that some of the long non-coding RNAs are a distinct genetic element in known lipid gene regions. So with the availability of more and more whole genome sequencing data sets in huge biobanks, there emerges this opportunity to analyze larger whole genome sequencing data for lipid levels. And we expect that larger number of samples with whole genome sequencing will allow us to identify novel loci at a genome-wide significance and provide some insights into the rare variants, and particularly non-coding rare variants. So what we did next is we have been analyzing the associations of lipid levels within TopMed, but now we're going to add in UK Biobank. And we're going to take a meta-analysis framework to now look at 268,000 individuals. So for meta-analyzing for GWAS, right, what we do is we collect summary statistics that give us an effect size and a p-value for each variant association with some outcome like LDL cholesterol, right? And we collect these summary statistics from each of the studies. And then we can create a pooled effect estimate and a pooled p-value based on combining the evidence across the studies and get a pooled value for each of the variants. 10 years ago, we developed a method to perform meta-analysis for rare variants that leverages score statistics and covariance matrices from each of the studies. And then we recapitulate the gene-based, the aggregation-based test centrally. And we showed that this approach was comparable to combining individual level data. And it is more powerful than combining, sharing gene-based p-values and doing a Fisher's approach or a min-p approach by the combining the study-specific p-values for the genes. And so here we're just showing that rare metal approach is more powerful than these other approaches for combining p-values. And this was the approach that I used with the exome-wide association study that I mentioned earlier. However, this rare metal method worked really well for exome sequencing data. It became computationally inefficient to use with whole genome sequencing data. And furthermore, it had a limited ability to be able to handle multiple functional annotations. And, as you saw with the previous TopMed study, we determined that actually incorporating multiple functional annotations is really helpful for looking at aggregates of rare non-coding variants. So we helped develop the MetaStar approach within the TopMed working group. And MetaStar takes the same framework as METAL by getting score statistics and covariance matrices, but it makes it more efficient by creating these sparse weighted covariances. And it allows us to implement the STAR framework, which allows the incorporation of multiple annotation scores. So we use this MetaStar approach with TopMed and UK Biobank whole genome sequencing data to perform individual level association analysis, as you would do in a GWAS, as well as gene-based and region-based association analysis. And with our substantially increased sample size, we were able to observe many coding aggregates and non-coding aggregates associated with LDL cholesterol. We additionally performed conditional analyses adjusting for nearby common genetic variants. And we note that some of the coding variants, as well as some of the non-coding associations, actually were no longer significant after conditioning on our GWAS variants. However, most of the associations remain, indicating these associations are distinct from the GWAS variants. We then looked at the overlap in signals between the coding and non-coding associations. And so just to orient you to this plot, on the x-axis, we have the signal from the association evidence for the non-coding signals. And for the y-axis is the association evidence from the coding signals. And we can see that some gene regions have signals for both coding and non-coding aggregates, where others are only significant for the coding aggregate test, and some are only significant for the non-coding aggregate test. One of the novel rare coding associations with LDL was ABCA6. This is a gene that spans 39 exons and encodes a protein structure of 1,617 amino acids. It has 2 ABC transporter domains and multiple transmembrane domains. And we found these robust associations, right, of these large sets of variants that are coding variants for ABCA6, right, associated with LDL cholesterol. So we found loss of function variants associated with LDL cholesterol, as well as missense variants associated with LDL cholesterol. And so what we did was we obtained the protein structure of the gene from the alpha fold repository. This had a 90% per residual model confidence score, making it a high quality protein to carry out some downstream investigation. And so we use this ABCA6 model structure. We wanted to look at the most important rare variants from our association testing, so our loss of function variants and our missense variants. So we used our annotation scores that we use within our statistical framework to identify crucial variants, which we define as having a PRED score greater than 40 for one of the annotation scores in either of these sets. And we identified 39 SNPs that we identified as crucial, we consider crucial variants with a high PRED score from the loss of function sets, and 30 from the missense sets, with a few that overlaps between the two. And so among the rare disruptive missense SNPs, there was a few that had these high mappability scores that identify variants uniquely and mapped to sequence reads. This includes 5 variants that were in the ABC transporter 1 domain. There was a few missense variants with high protein scores that directly reflect protein structure, but these were spread across the protein structure and had missense mutations comprising of cysteine and tryptophan amino acid changes, so they might not be that damaging. However, we thought this approach of using a modeled protein structure is a way that allows us to see whether crucial variants, right, from our association tests are in particular sections of the protein structure. So to pull it all together, right, I said at the beginning where we use GWAS summary statistics as a tool to further understand the biology of a complex trait, such as blood lipid levels, through using these secondary computational approaches with GWAS summary statistics. We also use whole genome sequencing data as a complementary approach for understanding the role of rare non-coding variants for blood lipid levels. We've been leveraging these two methods, STAR and MetaSTAR, which are methods for aggregating rare non-coding variants. I do want to note that most of our rare associations at this point to previously known lipid GWAS loci. However, our results are suggesting that this is an additional type of variation that's distinct from our known lipid GWAS and gene-based associations. And what you saw through the whole genome sequencing studies we've been doing, that really large sample sizes are necessary to make new discoveries. And getting back to the overall goal I stated at the beginning, right, we're looking at the spectrum of variation, genetic variation, and how it contributes to blood lipid levels. These are the key TopMed lipids investigators. Margaret has been key in analyzing the TopMed whole genome sequencing data and then did the meta-analysis with UK Biobank. Yushin is a PhD student who worked on the long non-coding RNA analyses. All this work has been done in joint with Preeti Natarajan, and Jerry Rotter is one of the co-conveners of the TopMed Lipids Working Group. And all of us work very closely with our counterparts, the STAR development team, in trying to develop STAR and make it useful for analyzing blood lipid levels. And then here are our key GLGC investigators that contributed to the series of papers that I presented at the start, looking at GWAS variants for blood lipid levels. So happy to take any questions at this time. I think there might be something in the chat.
Gina, I think the chat question referred to your FEWA slide from early on.
Yeah, so the FEWAs that we did, I can go back. We didn't do an false discovery rate, but we did there. I think there's a line there where there's a lot associated phenotypes associated with or different traits associated with the genetically predicted LDL cholesterol. And we did do a correction for the number of traits that we looked at. So you can see here we have this red line, which is a Bonferroni correction for the number of traits. And so you can see several other traits are significantly associated with predicted LDL cholesterol beyond the ones I highlighted.
So Gina, thanks for a really amazing talk. This has been a real tour de force. Do you think it might be possible, given what you've done here, to be able to turn around and say, hey, our rare variants have pointed to these non-coding regions, and basically predict that other rare variants in those same regions right nearby may be equally impactful, so that you can take a person's genome now and their rare variants, even though they haven't been studied elsewhere, by proximity to ones that you've looked at, they're actually potentially predictive.
Yeah, that's a great question. I think, you know, we'd have to look and see what is the the annotations that overlap between them to try and make a better prediction on which of the rare non-coding variants might be functional. I think not just proximity, but using some of that annotation information on top of that might help to predict the variants that are functional in a new population, right? That have novel variants that we don't see in our study. The other thing to say about that is when we do these aggregate-based tests, not all variants within the aggregate are going to be functional, right? So we're looking at it in terms of a whole and hoping that, we can use something like those alpha folds to protein predictions to try and see if variants are grouping together. That's gonna maybe be a clearer sign that a set of variants might be in a functional domain of the protein structure.
Yeah, and I have a second question. So I have to ask about CDKN2B AS1 in lipid levels. Has it ever shown up at all? Because that's a gene that's come up in heart disease, diabetes, glaucoma. It just shows up in so many different GWAS. And I'm curious whether your lipid level studies pointed to CDKN2B or CDKN2B AS1. So cyclin-dependent kinase inhibitor 2B.
Is that the chromosome 9?
Yes.
Yeah.
Yep.
I don't recall.
Okay.
It is not on, the list of those, it's not on that list of genes that we have that we are clear signals for lipids, right? Those 35 genes or the 25 genes that we know affect lipid biology. I don't know if it comes up in our GWAS. That's a good question.
Great, good. Thanks. Thank you.
Gina, question about your non-coding rare variant the link RNA rare variant association. Are there any EQTLs for those link RNAs that could be used in place of the rare variants to help prove causality in the sample sizes available?
Yeah, we really focus on the rare variants, right, for the long non-coding RNA analysis. And that is because using common variation, you run into the problem you run into with GWAS is LD, right? And so variants in LD, you'll have a variant that is in a long non-coding RNA but it's also in LD with another variant that is in the gene. So how do you tease apart, right, those two variants of being, if they're in LD with each other? Is it the gene or is it the long non-coding RNA? And so that's why we focus on the rare variants to remove kind of that linkage piece of for the long non-coding RNAs with common variants. And if we had a large enough study of expression data, right, in individuals that we have for the sequencing, right, then we would see whether the expression changed for the rare variants. But the problem with overlapping the rare variants with the expression, it has to be in the same samples because you're relying on there being that variant in both sets, right? And so you have to have, and that's just, we don't have that much expression data as we have whole genome sequencing data to make those insights. So, yeah, I've been saying we need expression data and everyone we have whole genome sequencing data for this very fact that I think that will really help us kind of understand some of the variation better.
So if nobody else has a question, I'll ask another question to me, but go ahead.
Yeah, I had just one more. The multi-ancestry analysis, a lot of, you guys in the lipids are always at the forefront, with the biggest sample sizes and in other fields, even common disease fields, we're trying to catch up. So I was curious, did you actually think it helps to find new genes or new loci, doing that multi-ancestry analysis, or are we finding the same things?
No, it helps on multiple fronts, which is in the Graham paper, which I only highlighted the polygenic risk score part of that. But you identify additional variants by looking outside just a European-centric GWAS. And it also helps with fine mapping, so you get smaller credible sets when you use a multi-ancestry GWAS as doing a single ancestry GWAS.
Great.
Thanks, Gina.
So Gina, my follow-up question, it's actually a different question. So Zetia, and Zetamide, so Zetia works to lower cholesterol by blocking absorption from the gut. And then the liver releases the cholesterol and you wind up with lower cholesterol in the long run. Statins have a completely different mechanism. So in my family, statins don't work on anyone. We have really high cholesterol in my family, and the HDL levels have always been very high. And as we get older, the LDL creeps up. And then the doctors want to put us on cholesterol lowering drugs. So one by one, everyone in my family has tried the statins. They don't work, and we wind up on Zetia. So that has made me wonder whether these different pharmaceuticals with different mechanisms of action might actually be predictable. Could you take a family and analyze what are going to be the drugs that will work for these people or this person?
I totally agree. I think we can, based on the data work, we haven't done any gene by drug interactions. That so far has not been attainable of a statistical approach to do. But you could think of ways to do that in terms of for the drug targets, the gene targets for each of those drugs, creating a polygenic risk score for those kind of different drug targets and predicting if someone has a high polygenic risk for that particular gene for the drug targets. As a way to kind of classify people into different subgroups that might be more amenable to different therapies. So, yeah.
It raises interesting questions when you see when you observe these things in your own family. And we've done 23andMe on all of us, so I'm happy to share the variants.
I think it's a really great question for what I do, leveraging all these data sets, right? We just don't have that kind of detailed information, right, on the medication to be able to do that kind of analysis. So you need a really uniquely or a collected cohort to be able to kind of tease apart those effects on the different drug classes.
Yeah,
it's a great way forward though. Yeah.
Well, if there are no more questions, then yeah, let's give Gina a round of applause and thank you.
Thanks everyone.
Thank you, Gina, for such a great talk.
I appreciate everyone attending and listening. Looking forward to talking with the students in a bit.